Guys, in this video we are going to cover DNA replication in prokaryotes. So DNA replication is going to have three basic steps. There is going to be initiation, there's going to be elongation, and there is going to be termination. So initiation, elongation, termination. One of the first things is going to be initiation. It's going to be the first step. So we have lots of different terms here on this slide. There are lots of enzymes involved in this. So the first one that we're going to mention is topoisomerase. It's hard to say. I actually got it out that time. Topoisomerase. It's a mouthful. So we said before that DNA is really tightly packed as chromatin. So it's wrapped around these histones. These histones are packed up really, really tightly as chromatin. And if we want to copy DNA, we've got to loosen that up and be able to get in there to attach the protein. So topoisomerase 2 is going to be the protein that comes along and loosens up DNA and allows us to then use helicase. So helicase is this blue wedge here. Helicase literally is a wedge. It's protein that's going to go in there and break those hydrogen bonds and basically unzip DNA. So remember, DNA had the hydrogen bonds that keeps the base pairs together. Helicase unzips the DNA. And now that we've unzipped it with helicase, we need to make sure that it stays unzipped. So we're going to use single strand binding proteins, these little guys here in purple, and that's going to make sure that they stay unzipped and that it doesn't come back together and the hydrogen bonds try and come back together and collapse together. So SSBP, single strand binding proteins, keep the zipper unzipped that helicase started. Then we want to start to lay down new DNA. To lay down new DNA, we're going to use our DNA polymerase. DNA polymerase lays down new DNA, but it can't attach without a primer. So we're going to need RNA primase to be laid down first. So here's our RNA primer laid down right there, and somewhere over here, we are going to have an RNA primer off screen that it doesn't show us. So there's an RNA primer that gets laid down before DNA polymerase can come along and lay down new DNA. So initiation was the beginning of it. We are going to have topoisomerase loosen up the DNA. Helicase wedges itself in there and breaks the hydrogen bonds and unzips it. The single strand binding proteins keep that zipper unzipped so they can't come back together and close. And then we want to lay down new DNA. So to do that, we need a primer. That primer is made out of RNA coming from an enzyme RNA primase. And now DNA polymerase, which is the protein that's going to lay down the new DNA, is going to attach. The DNA polymerase is then going to lay down new DNA. So now we're into step 2, which is elongation. DNA polymerase lays down new DNA. There is an important fact that you need to know about DNA polymerase laying down new DNA. And when it lays down new DNA, it is going to have to lay down DNA in a 5' to 3' direction. So 5' to 3' always. So DNA polymerase, here's our RNA primer. And now we are going to use that as an attachment point for DNA. DNA pol 3 attaches on, and it's going to run down like a train, just laying down new DNA, laying down new DNA, laying down new DNA. And it runs, again, 5' to 3'. That's how DNA pol lays it down. So it begins on the 3' end of the old, or the template, the old DNA, because we said Messelson and Saul said that this was semi-conservative and that we split it apart and half of it's old and half of it's new. So the 3' end of the old DNA is going to have this hydroxide end on it, and we're going to bond our RNA primer and our DNA pol, and it's going to start at that end and continue 5' to 3' in this direction. So on this strand, this is called the leading strand because it can attach in one place and just keep on trucking. It doesn't have to stop. It can just lay it down 5' to 3' like it wants to. So the leading strand is awesome. It's just one continuous piece, super easy. The lagging strand, however, is not as easy. The lagging strand is going to be in a different direction. So we need to create that one in pieces, and those pieces, here's one, those pieces are called Okazaki fragments. Okazaki fragments, and they are, again, DNA polymerase laying down DNA, but it only lays it down 5-3, so it has to do it in fits and starts and chunks. And those chunks are called Okazaki fragments that are found on the lagging strand. So Okazaki fragments on the lagging strand are laid down. Then we are going to have DNA pole 1 that's going to, after we take, we need to make sure this lagging strand is going to be all DNA together as one piece. So with the lagging strand, we are now going to remove those RNA primers on all of these places. So DNA pole will help remove the RNA primers, and ligase is going to be our glue. So here's our ligase, our glue to put those chunks together. So Okazaki fragments here and Okazaki fragments here, and ligase is going to glue that down and make sure that this is one continuous strand of DNA. So now the lagging strand will look just as pretty as the leading strand when we're done. At the end, topoisomerase 4 is going to help make sure that it is clear and good to go, and we are at the end and it's ready to terminate the replication. So these are our 3 steps: initiation, elongation, termination. And at initiation, topoisomerase loosens it up. Helicase is the wedge that splits it and unzips the hydrogen bonds. The single strand binding proteins keep it split so they can't go back together. RNA primase lays down the primer. DNA pole is going to lay down the new DNA. That's going to lead us into elongation as it lays down new DNA, and it's going to lay it down at the lay it down from 5' to 3'. It needs that hydroxide end at the 3' end of the old DNA where the primer is. So they're going to lay down the strips. The leading strand is good to go. The lagging strand is going to be in bits and pieces called Okazaki fragments. We are going to remove all the primers and put them in, put DNA in its place, and now we are going to glue them together using ligase, which is our glue, and that's going to bring us to termination, the end of this process. So we have all of the different enzymes here in this table that tell you what they're doing. Definitely know DNA pole lays down new DNA. Helicase is our wedge. Ligase is our glue. We can't lay down new DNA without a primer from RNA. Our single strand binding proteins are going to be used after helicase splits it apart. They're going to make sure that the DNA doesn't bind back together and rewind. And then we've got our topoisomerase. 2 of those, one of them is going to make sure that the supercoiled DNA relaxes, and then the other ones are going to make sure that we will reseal the DNA at the end of termination. So this is DNA replication. Let me know if you have any questions.